Welcome to the third lecture of the third week of this course, Human Molecular Genetics. In the previous two lectures, we have looked at two distinct group of mutations. One you call as a gain of function mutations, other one is a loss of function mutation. And in the gain of function mutation, we ended with a note that it is often difficult to understand how a given mutation changes the function of a protein in terms of the function of the protein in the cell, tissue or organism, right. It becomes very challenging because you do not know the function of the protein. So, then if you know the function of the protein, then you can ask how a mutation altered the function of the protein. So, for such kind of approaches, it is going to be very extremely difficult to study humans. So, we need models. We need models, meaning you have to use some other organism and create the condition wherein the organism also has got a defect very similar to what you see in human, and then ask now question what goes wrong in this organism and study what are the events, chronological events, which happened first, which happened second, and so on, and then try to understand, and this is the physiological pathway, and this is what it is abnormal, and this is where the genes function, and so on. So, that is where the model system for human disorders become very, very important. So, that is what put on the slide here. Often the disease pathology is progressive, meaning to begin with you are normal, like we have seen Huntington disease. You have different generation to begin with, the individual is normal. At certain age you have symptoms that become severe and severe, and therefore it is very challenging to study it in human, because to begin with you do not know that individual is going to have the disease or not. Secondly, even if you know, how are you going to really look at what happens inside the human body? You cannot do any experiments with the human, which is ethically not allowed. And second, for many of the genes that we know that they are defective and results in the disease, we do not know the function. That is something that we have already discussed. So, you need to understand. So, here we use animal models for the human disorders, and such kind of study using the animal models have contributed enormously to understand the pathophysiology, what goes wrong and what is the function of the protein. And with that we also try to go for some therapeutic attempts. Can we reverse the phenotype, right, and that can have medical implications in humans. So, really the animals really help us to understand the biological functions of genes and their protein and how it affects results in the disease condition. So, simply because humans cannot be experimented, we only look at individuals that have had normal mutation, meaning natural mutation, and we try to study them and try to understand as much as possible. So, what are model systems? So, the model systems are, when you talk about model animals, these are particular species of animal developed over many years, because you have to choose and you have to characterize, understand how you can maintain him in the lab. You cannot go and take a wild animal and then study on that, because they are like humans. There are a lot of variations, and whatever effect that you see at the end of any experiment in a given set of individuals of organisms is going to be very difficult to infer like humans, right. Therefore, you need to have lab bred animals. So, that takes years to come up with certain system, and these are experimentally powerful, very powerful to answer some of the very important questions with regard to the disease biology, disease pathology, or even understanding how a gene functions. Now, what is important is that not all models are good for all questions. The models are as good as the question. So, you need to know what question you have to ask with what model system. You are talking about cognitive ability, let us say you are talking about how the brain functions. Now, these kind of questions you cannot go and ask using a cognitive function very, say, unique to mammals or unique to primates, because the way the brain is formed, and you cannot go and question it using some other model system which is not even vertebrates. It becomes difficult because the way the brain is organized is different and so on. So, one need to know what question you are asking. Accordingly, you have to identify a suitable model. I am listing here some of the model systems. People popularly use it for many biological questions. For example, Aplysia, this is a mollusk, a soft bodied animal. which is a kind of a model which really helped us to understand the neurophysiology, right? So all the information that we know about how the neurons function, for example, the aplesia's ink release response, right? Because there's a signal that releases ink. All this information have come from this model. It's very, very powerful in neurophysiology. You have another model which is the C. elegans, a nematode, a worm, which is a very good model for understanding the development and physiology. And as you know, many of the model systems have led to discoveries that were awarded Nobel Prize. Every model you can count on that they contributed to the Nobel Prize. That's the significance of their contribution. You have zebrafish again, which is a vertebrate. They have transparent body, and now this is a very, very powerful model system to understand the metabolic process. People are trying to model them for diabetes and obesity and many, many different conditions. And you have the fly, fruit fly Drosophila melanogaster. Again, it's a very, very powerful species for genetic manipulation, for developmental biology. They're very, very good. And in all these models that are there in the except the aplysia, you can engineer, you can change the genome, you can make the change in the genome the way you like, so you can model them beautifully. The models that belong to the vertebrates that are very close to the humans are the chicken, for example, Gallus gallus. These embryos are very, very good for developmental biology. Majority of our understanding with regard to how the embryo grow, what are the signals that contribute to the embryonic growth, how come from studies from chicken. And then you have rat, the lab rats, and they are particularly useful model system for many of the neurophysiology, toxicology, for example, any of the drug safety or all these things. People use this model system to see that the compound is good for human applications. So they are very, very good, especially for neurological studies. Rat has been the most popular model because the brain size is bigger. You can investigate different regions of the brain. But when it comes to human genetics, modeling the human disorders, the most favored model is mus musculus or the lab mouse. You must have seen the small white mouse that people use in the lab studies. These are cute animals, easy to handle. They don't really bite you. And important thing is that you can change their genome. You can change their genome the way you want. Therefore, you can model it for a given human disease, right? That's very, very powerful. And majority of the human genetic disorders are modeled using the mouse. So we are going to look into some of the approaches people use to model mouse to understand human genetic disorders. So we'll take maybe 2 or 3 approaches. We will not get into all the approaches. We'll show some examples as to how that has been used to understand the disease biology. Let's look into 2 distinct approach people use when they use mouse as a model system. One is called as transgenic animals, meaning you are putting some extra copy of DNA, and this DNA need not be from mouse. It could be from the human, or it could be some engineered DNA sequence. You can put it inside the mouse genome and let that foreign DNA express and give a phenotype that you want the animal to develop. So these are as I told you, exogenous DNA inserted into the germ line. What do you mean by germ line? These are transmitted via the germ cell, so you can maintain a line, you can breed them, you can maintain the line. And the DNA has its own regulatory element. So when you put a DNA, it has its own regulatory element, meaning it has its own promoter. So it would tell you where it should express, when it should express, what should be the quantity of expression, and so on. And that drives the expression. Often such models, that is transgenic, when you put an extra copy of the DNA inside the mouse genome, they represent what is called as gain of function mutation. Remember, we discussed a gain of function mutation, like for example, Huntington disease. You have the CAG repeat. When the repeat length goes beyond, for example, 40, 50, 60, and so on, you end up having disease. Now you can insert a gene sequence having expanded polyglutamine, and this is a gain of function. If that gene is expressed, if protein is made, you are going to have the symptoms. At least you can expect that to the animal to have that. You cannot model loss of function using transgenic. It is not impossible, but it's difficult. The reason is, for any gene that we talk about in the human, you have a homologous gene in the genome. So even if you put an extra copy which is representing a loss of function mutation, it is not abolishing, or it's not competing with the normal gene that the mouse has. So it's not going to really express that. So that's a difficulty, right? So normally you're going to have extra DNA, like you can model beautifully Huntington disease. You can put a gene that has got expanded polyglutamine coding region, or you can even model, for example, conditions like Down syndrome, where you have 3 copies of certain genes. So you can pick those genes that are 3 in copy in the human, and we can make a transgenic animal in the mouse, and that may develop a Down syndrome like phenotype, and you can study why your Down syndrome, when you have 3 copies of certain gene sequence, you have mental retardation. So you can model the animal and see is there anything can be done to rescue the phenotype. So that's about transgenic. So you have the other group which is called the knockout, which is the most powerful approach you have. You have another approach that has come recently called CRISPR-Cas gene editing. We'll not get into that, but this is something which is well established. You have it in many of the textbooks, so we'll restrict our discussion to the knockout. So what is that knockout? So basically you try to inactivate or modify an endogenous copy of the gene. So the mouse has got its own gene, so we are going to remove the gene or its function or alter the function of the gene to mimic what you see in the human. So you don't put any extra copy of DNA here, copy of gene here, but you change subtle changes you bring about in the genome of the animal that results in the condition, right? So that you can model both gain of function and loss of function mutation here. For example, if you talk about a missense mutation, that could result in a gain of function or a loss of function. You can by using this approach, you can even create such kind of a subtle differences in the genome. So now a missense mutation could be loss of function, you can model it, or it could be a gain of function, again you can model it. So that's the approach of knockout animal, a variant of which is called the knock-in, which is used for it, this kind of applications. But we will be talking about a knockout which is generally for loss of function effect. Let's see transgenics. How do you put an extra copy of DNA inside the mouse and make it to express? So what do you need? You start with what is called as a construct. The construct is given here. So what is this construct? This construct has got an element which you call as a promoter, right? So what is promoter? This is the element which informs the DNA or the gene as to when it should express, where it should express. So you may have a DNA binding protein, which is a transcription factor, comes and binds here. As long as the transcription factor is there, this gene will be turned on, so you will have the protein being made. So what do you put here? So this is the coding sequence. So often the coding sequence is derived from cDNA, because your gene in human could run into several kilobases, hundreds of kilobases. One of the genes even go up to 1 megabase. So you have multiple exons, 10 of them, 20 of them, spreading large segment. So that such big DNA to extract and put inside is going to be extremely difficult. So what people do is they take the cDNA, which is the processed DNA, meaning it has all the exons joined together. So it has the coding sequence. You fuse it with a desired promoter and make a construct. So the cDNA would express, it will make the same protein. Otherwise, the endogenous gene would make, right? What are the promoters? The promoters could be the endogenous, meaning you use the human promoter. The gene that you got from human, obtained from the human DNA, you can use it with its own promoter. It may express, right, if it has got all the elements. Or you can use a viral promoter. For example, the virus has got its own promoter, which would function because it uses very few transcription factor that we make. So they are able to successfully make their own protein. So we can use those promoter fuse to whatever sequence that we want and then make a construct. So, now they will express very high, or we can use a promoter of a gene of mouse origin to drive your transgenic expression only in certain tissues. For example, I want my, for example, the Huntington protein, which is having the expanded polyglutamine stretches, to express only in the neuron. So, what I will do? So, I will go for any gene whose expression I know is restricted to the neuron, take that gene promoter, fuse it with, for example, my cDNA coding for the Huntington. So, what would happen when you make a transgenic? Although this particular gene copy would be present in all the cell, it would express only in the neurons, because it has got a promoter of a gene that expresses only in the promoter. So, this is the way I will be able to control the expression, right. So, DNA, how the DNA gets integrated. So, when you inject the DNA, we will come to that, how it is done. So, it may get integrated anywhere. You do not really tell that this is the place in the genome you have to go. So, it is random, it goes and gets integrated anywhere. Now, the expression of the gene copy that you put in depends on its site of the integration. There could be regions, for example, centromere region, where the spindle comes and binds and pulls the chromosome apart during cell division. And these regions are not gene rich. Normally, they are very condensed. If your DNA gets in there, it may not express. Or it could be a region which has got many genes. So, if it gets integrated, it may express. While doing so, it may also disrupt the function of a gene that is located there. For example, it can go and land in a region where there is a gene, the transgene is expressed, but your normal gene that was present there could be affected. So, you have to look at several lines there. So, that is what the last point is, integration might affect an endogenous gene. So, it happens. So, you have to look at several animals and select one that is really good. So, that is the disadvantage and advantage of transgenics. Let us see how we do it. So, what you do is that you cross a male and female to get embryos, right. Now, these are embryos in which you have still they are single cell zygote. You have nucleus from the egg and the nucleus from the sperm yet to fuse. So, that is the stage at which you integrate the DNA that you want. So, you basically set the crosses and then you anesthetize the female and then take out all the embryo that are just fertilized, but yet the nucleus, the male and female nuclei yet to fuse, and that is the stage in which you use a very thin capillary kind of needles to introduce your DNA, pretty much inject the DNA inside the nuclei, right. So, when you inject, allow it to grow for some time. In this process, when the male and female nuclei fuse together, there will be opening up of the genome, and during this process this DNA may get integrated somewhere, and then you take the embryo, put it into what is called the foster mother. The mother is not the biological mother for the embryo, but you condition her and put the embryo inside and allow her to deliver the pups, and you have the animals that are born, and some of them would carry their DNA in every part of the body, including their germ cells. If it so happens that some of the germ cells of these animals have this transgene, if you cross them, then they are going to go to the next generation, and you can characterize. So, how do you know these are derived from this embryo, not from this mother? Because you can use the fur color. For example, white is the foster mother, her fur color is white, but the embryos are derived from a brown or black mice, and if the pups are black or brown in color, you know that they are derived from this embryo, because this coat color is dominant over white, and there are no white here, and that means that they are not from this foster mother. So, this is the way you characterize the transgenic animals. So, that is how you use the foster mother to create animals that harbor the transgenic genes that might express some foreign protein. So, what you are going to see is an example of how a transgenic can be a powerful tool to express a protein that are not native to the animal. So, what is shown here is 3 animals born to a foster mother, and the one on the extreme left and right are the so the one on the left and right are the animals that express a foreign protein. This protein is called as green fluorescent protein or GFP. So what is the property of the protein? So when you excite these protein with certain wavelength of light, they fluoresce, they give you fluorescent color, and that's what's shown here. So you can see that the animal on the left and right, they have green color in their ear, in their tail, in their near their nose, and then eye. You see that this glittering green color, whereas the animal that is in the middle is not showing such green fluorescence because it doesn't have this particular gene. So it clearly tells you the power by which you are able to express a protein which is a native protein of a jellyfish, a marine species present in seawater. That gene you have obtained, you have used that, engineered it, and you are expressing it in animal, and it expresses the protein which is otherwise not present in your in your cell. So that's the power of the transgenic animals. So you can, for example, express Huntington protein with expanded polyglutamine and see whether it develops disease like Huntington disease, and that's what's shown here. So here what we are seeing is a transgenic mice wherein you have added extra copies of the gene having expanded polyglutamine tract is now expressed, and then you are comparing it with a wild type which is a normal animal, and this is the animal that expresses the mutant Huntington protein, right. So what you can see here is that when you lift the animal using a tail of the animal, then the animal normally try to explore what is around, try to escape, but when they have a very poor coordination of their hands, limbs, and what do you call hands and legs, then they try to clasp. This is called as a clasping phenotype. So when they don't have the motor coordination ability, they clasp like this. This is one of the classic phenotype of motor deficit in motor coordination. So this is an animal that develops Huntington phenotype. Now why you should develop animals? So it really helps us to understand what happens at the level of cell or neuron and why you have the disease. So what is known now is that by studying such kind of animal models, the polyglutamine protein form aggregates, and these aggregates recruit a large number of proteins that are otherwise required for the normal function of the cell. As a result, the neurons don't function properly and they slowly die, right. If you are able to clear these aggregates, then the neurons function better. So what is known is that is there a way by which I can help the neuron to overcome such kind of toxicity, stress, and people have used various ways, and this is one such lead molecule a group has identified when they have treated the animals that are expressing again the mutant Huntington, and you can see that they are able to behave the way just like the wild type behaves. So this animal model helps us to come up with some therapy, at least in principle, a kind of a proof of concept approach using the animals. You can screen, and then you will be able to take it to the human applications. So that's where the model animals really help us. We understand how a defective protein results in the phenotype, and we can also come up with certain ways to eliminate the abnormal function that a gain of function protein has offered to the cell wherein it's expressed. So that's the power of these animals. Now we are going to go to the next approach, what you call as knockout mice, wherein we generally remove the function of a gene that you are looking at, normally to generate a condition which is a loss of function, right. So let's look into how do you make the construct for the knockout, for generating knockout mice. So the way you are able to disrupt a gene function in the knockout gene approach is by homologous recombination. What do you mean by homologous recombination? So you identify a region of a gene which you want to disrupt or delete or inactivate. So which region you would identify for such kind of deletion? So So, it depends on the gene that you are looking at, the information that you have about the gene. One safe bet would be to delete the first exon, because the first exon would have the promoter sequence. So, if you delete the promoter sequence along with first exon, the gene is less likely to be expressed. So, you pretty much brought in the loss of function mutation. If you are not sure about which is the first exon, you can go about deleting another exon. The reason being, at times certain genes could have more than one first exon, different promoters. Even if you delete one, the other promoter may still functional, therefore you may not see the desired phenotype. Therefore, you want to go for certain exons which code for the functional domain of the protein, let us say a transcription factor. So, what is the functional domain for that particular protein? It could be the DNA binding domain, because a transcription factor needs to bind to the DNA for activating the gene for its expression. So, if you know the exon, which exon codes for the DNA binding domain, you can decide to delete that particular exon. So, let us have a look at here, how do you really do this. So, here in this particular cartoon that we are seeing an exon which codes for, let us say, DNA binding domain. So, I have decided that I would knock out, I would delete this particular exon. So, what I would do? So, I will go for the so called genomic DNA library for this particular strain of mouse that where I am using, and identify the fragment that represent this region of the gene. Likewise, identify the fragment that represent this region of the gene, right. How do I cut these fragments? I use those enzymes that you described, that is restriction enzymes, which would cut the DNA at desired region, and you are going to use another protein which you call as ligase, which join the DNA fragments. So, we use these to identify and clone a small region that represent these two segments of the gene. What I do? I fuse these two segments with another segment which is not from the mouse origin. It is a synthetic construct which codes for a protein called neomycin resistance protein. So, what is the function of this neomycin resistance protein? So, this protein codes for a protein which is able to degrade the antibiotic called neomycin. So, in the presence of neomycin resistance protein, the cell can grow even when you have neomycin there. So, in the absence of this protein, the cell cannot grow, it will die. So, why this is important? We will come back to this little later. So, what you have done is that this whole thing is put in a plasmid. We remember we discussed that these are circular DNA which is present in E. coli and so on, which is used for the cloning purpose. So, you have used this, and what you do is you use a restriction enzyme, cut here, you make it linear DNA and use it for transfection. What is that? You introduce the DNA inside the mammalian cells derived from the mouse. What cell type will come back a little later. So, you can introduce that. So, this DNA get in there, and once it gets in, you expect a process called as homologous recombination, something that is shown here. So, you may expect a homologous recombination happening here and here between the homologous sequence, just the way we discussed in meiosis recombination. In somatic cells, although very rarely such events happen. So, if that happens, what do you expect? You expect if there is a double strand break here, double strand break here, double strand break here, double strand break here. So, this DNA would get joined with this and this, and you will have this DNA get joined with this and this. As a result, you may have a plasmid having the exon of the gene that coding for the DNA binding domain, whereas your gene now has got the neomycin. So, your gene would look like this. Now, in the exonic region, you have the coding region for neomycin resistance. So, once it gets in, you grow the cells in the presence of neomycin. Now, all the cells that carry this DNA fragment would survive in the neomycin. They will multiply, grow. Those who do not have such neomycin resistant gene, even if they contain it in the plasmid like this, it will be degraded very fast within couple of cycles, and then the cells would die because they cannot survive in the presence of neomycin. But what is this is called as positive selection. You select for a particular phenotype, that is resistance to neomycin. But this kind of integration, that is the integration in exactly the region that you want that to happen, is a rare phenomenon, right. It can also happen by random integration. For example, the DNA gets integrated somewhere in the genome and still the neomycin resistance would be there. The cells would code for that protein and therefore they would grow. So, how do you know whether the cell that survive under neomycin resistance would have the same kind of integration that you desired? So, what people do is they go for what is called as a double selection. So, when you make a construct, for example, this region you add another coding region. Now, this coding region codes for a protein which could be a toxin, meaning if this protein is expressed, the cell would die. So, if the DNA like having the toxin and the neomycin resistance gets integrated anywhere in the genome, then that would result in the death of the cell because the toxin is expressed and therefore the cell would die. But if the integration took place because of what is called as the homologous recombination happens here, now the neomycin would replace this DNA binding domain coding exon, but in that process the toxin will not be get integrated, or the coding sequence for the toxin will not be getting integrated into this genome. As a result, they would not die because of toxin, because that is not expressed, because that is excluded, that remains in the plasmid and gets degraded, right. So, this is called as a double selection. You select for a phenotype that is survival and with neomycin, and you also select for another phenotype that is toxic. If it gets integrated in the genome, then which is not a homologous recombination process, the cell would be eliminated. So, in this process you are able to get groups of cells wherein such kind of exchange, the exchange of the DNA binding domain coding exon with neomycin resistant cassette could happen, and that would have an allele in which the exon is deleted. So, that is how you generate cells in which you are able to create the mutations the way you want, right. So, that is the first step. So, what do you do next? You have to create the animal. So, how do you create the animal having this mutation? So, it all depends on the cell in which you are making such kind of change in the genome. So, normally for generating knockout mice, people use what is called as embryonic stem cells. What are they? Because these are called as stem cells because they have the potential to develop and differentiate into any cell tissue type in your body, because these are embryos. Every cell has the equal potential to develop any part of your body. So, that is where you derive. So, in this case what happens is that the egg and the sperm fertilize, and you have a stage of embryo called as morula, and then you have a stage called blastocyst. The blastocyst stage you harvest these cells and cultivate them in the culture dish, and which you call as embryonic stem cells, because they have all the potential. Now, you can culture them in the lab, and then you transfect them with the construct that we discussed, allow such recombination to take place, and then you identify cells that wherein you have had such recombination took place. So, you have created the mutation, right. So, once you have that mutations created, you can generate animals. How do you do it? That is what is shown here. So, as we have shown here, we have a construct in which you have a neomycin gene, you have transfected using a process, here it is shown as electroporation. There are more than one method, wherein you expect this kind of homologous recombination which replaces the exon of a gene with the neomycin resistant gene. That could happen in one of the cells that you have seen here because of the selection, either because of or both because of the antibiotic resistance and because toxin not getting integrated into the genome. You would allow only those cells in which such integration took place, and they are preferentially grown in a plate. So, you would have now all the cells having the kind of change that you expected in the genome. Once you have grown enough, then you take few cells, these embryonic stem cells, wherein you have confirmed that you have altered the genome, and inject inside the same embryonic stage. If you remember, we have discussed about blastocyst. This is the stage from which you have taken the embryonic stem cells. Now, you can put the cells back into the same stage of embryo using a fine needle. So, still you have some space here. Normally people add some 12, 13, 15 cells inside, and once allow, then what happens, these cells get integrated inside the embryo, they grow and form different organs. Eventually that embryo is put into a foster mother, the mother who is not biological mother, but put inside her uterus, allow it to grow, and then you have the embryos coming, right. So that's, the embryo comes in. That's what is shown here. So what you, just to summarize so far what we have discussed, so you have a neomycin cassette which you use to, for example, remove certain exons. Here what is shown here is more than one exon, exon 1, 2, 3, and then you have put that inside the blastocyst. You have derived the embryonic stem cells, you have put that blastocyst having these embryonic stem cells inside a foster mother, and then these embryos grow. So in these animals you would have some tissues, some part of it would be derived from the embryonic stem cells in which you have changed the genome. So that's what's shown here. For example, if you have derived the embryonic stem cells from a mouse strain that has given you brown color, coat color, and you have put that ES cells into a blastocyst of animals that have the white fur color, then you would have patches of your skin having brown patches. That shows that it is a chimera, meaning it has the cells that are derived from the embryonic stem cells that you have engineered. Now you cross it with, for example, a white color animal, and you look at all those animals that are brownish in color because that is dominant over the white. That would indicate that the germ cells that gave this phenotype is derived from the embryonic stem cells that you have put into the blastocyst. So that's the way you identify an animal which now carries 50% of the genome coming from the embryonic stem cells that you have derived. Now this has got 50% chance that it carries your knocked out allele. Now you can cross, for example, a brown animal with the other, which are supposedly heterozygous, and you can get a homozygous animal which has got both alleles, in which the exon that you wanted to be deleted is deleted, and now you can study the phenotype, right. So this is the way you engineer, you delete a certain exon and look at the phenotype, right. Now when you do this, when you do such approach, when you want to delete, in this approach what we had discussed is that this deletion happens in all the cells that animal carries, because you have deleted it in the embryonic stem cell itself. So every tissue that is derived from embryonic stem cells will have the deletion. But what is the bottleneck is that if the gene that you are studying has got more than one function. One of the function could be, for example, the early embryonic development. The other function could be something very unique to, for example, the brain function, neuronal function. So if the gene has got a function in the embryonic development, if you have deleted that, then the embryo is not going to survive. So you will never be able to understand the function of this particular gene in the neuron. So what you should do in that case is, you should allow the gene to be present during embryonic development, but delete only when the embryo becomes an adult and then you have the neurons differentiated, only then you want to delete and then look at the function of that gene, right, in the adult. So that is, this approach is called as conditional knockout, meaning you want to delete the gene in a particular tissue at a particular time point during development. So that is called as conditional approach. It is very similar to the global knockout just now we described, except that you don't inactivate the gene in the embryonic stem cells. So you sort of construct the targeting construct such a way that it does not disrupt the function of the gene. Still it is able to code for the protein, but it has some desired change which will use it to delete the gene in a given tissue at a particular stage of a development. So this is called as conditional knockout. How do you do this? So what you do in this case is that you create a construct, right, which has a unique site called LOXP site. These are a few bases of sequences which are recognized by a recombinase called Cre recombinase, and this recombinase, if it is present, it is going to identify these LOXP sites and promote what is called as site-specific recombination. So how does it happen? For example, in this allele what we are seeing is that we have introduced 2 lock speed sites flanking exon 1, 2 and 3. So, what you are trying to do is when lock speed sites are present there, the gene is still able to function, because here between exon 3 and 4 this lock speed is present in the intron, therefore, it will be spliced out when the transcript is being made, still the gene is functional. However, if you express the Cray recombinase in that particular cell wherein you have this engineered copy of the gene, then the Cray recombinase would favor a recombination called as site specific recombinations. What is that? So, you have this, you have the lock speed sites, and let us say you have the 3 exons, another lock speed sites is there. So, what happens is that because the sites are directional, it can form this kind of arrangement, and your 3 exons are in between here, and the recombinase would make a double stranded cut here and switch them. As a result, what could happen is that you would have the DNA, this to this gets joined, and all the intermediate region having all the 3 exons will be spliced out. Okay? So, this way you are able to see that. So, that is what you are seeing here. So, you are going to cross an individual animal in which you are able to put LOXP on either side of exon 3 in the embryonic stem cells, selected the embryonic cells, and you made the animal. Now, in the animal genome you have this. Now, you cross that with a transgene, this different method that we discussed earlier, wherein you are expressing a Cray recombinase. Now, the question is where the recombinase would express. I can use a promoter which drives the Cray recombinase only in, for example, astrocytes. These are supporting cells in the brain, for example, or I can drive the Cray recombinase coding region with a promoter of a protein normally expressed in muscle or heart or liver. So, it all depends on where I want the recombination to take place. So, in this case, we have crossed the animal which expresses Cray recombinase in astrocytes and crossed it with an animal which carries the LOXP sites on either side of the 3 exons of a gene that you are interested in. As such, this animal is normal, right? That is what we discussed, that LOXP that you put in such a way that it does not affect the expression of the gene. The moment you cross them, what happens? In the astrocyte, in this particular cell type, you have the Cray recombinase which favor this kind of a recombination, leading to the elimination of all 3 exons. Now, the DNA from here to here will be joined, this is lost. So, that is what is shown here, right. So, you have had condition wherein in the astrocyte the Cray recombinase is expressed and it has deleted all 3 exons 1, 2, 3, but exon 4 is present. That does not really help because the gene cannot express. Whereas, in other cells wherein you have no Cray recombinase, this LOXP site is intact, it does not change the function of the gene, and the gene is happy, the cells are happy. But if you are seeing a phenotype in this animal, that means that this protein is essential for the astrocyte function, or if you want to likewise model it for liver, you can model it for muscle and so on. So, that is what you call as conditional knockout. You want to delete or disrupt a particular gene in a given tissue or in a given stage of development, depending on what question that you are asking. So, that is precisely the way people remove the gene to understand the tissue specific function of the gene. So, what I am showing is one example of conditional knockout, wherein they were able to identify a function, neuron specific function of a protein which otherwise was not appreciated much. So, this protein called as methyl CpG binding protein 2, MECP2 gene, if this gene is mutated, then there are a large number of individuals show a mental retardation called Rett syndrome. This is one of the very common mental retardations, and more often this disease affected the female. There are many patients that are the children that are female, right, very few boys, and one of the hypotheses was that if you have this gene defect in children that are XY, it could be embryonic lethal, right. This was the hypothesis. So, what and it was very surprising because this is a methyl CpG DNA binding protein, because it's a transcription factor, and they never thought it could be having such a tissue specific function or neurological function. They thought it was a generic transcription factor involved in all the tissues because expressed everywhere. So it was a surprise when people identified mutations in this particular gene. And before that, there were papers wherein they are shown if you knock out this gene, remove the entire copy, even the embryo don't survive, right? So maybe in these individuals who develop Rett syndrome, the mutations are such that it is a leaky protein, meaning the function still is intact, not to that extent that you see in normal, but still some function is left. Therefore, you are seeing, they survived during development, but later they show neurological, mental retardation and so on. So they wanted to model. So you create embryonic knockout, then what happens? Embryos don't survive. How would you study the function of the protein in the neuron? So this particular group of this paper, if you are interested in, you can go and read. They developed what is called as a conditional knockout, wherein they deleted the gene only in the neurons, differentiated neurons, by using a Cray recombinase is expressed only in the differentiated neuron, and they were able to model and show this animal which had a deficiency of this protein only in the neuron, showed many of the phenotypes that are seen in Rett syndrome. One of them being that the size of the brain is very, very small as compared to the wild type. So you can see that all the unfilled white circles are the tissue sample representing the brain of wild type animals. So with the age, and if you look at the brain weight, you see that consistently the MECP2 deficient animals showed smaller brain, and they have had many stereotype behavior and other things that typical of Rett syndrome. And there are even now lead molecules people have identified using the animal model to cure possibly the Rett syndrome. So that's the power of the animal models, the approaches people have developed. In fact, for the knockout animal, the approach that they developed, this approach led to the Nobel Prize, right? You want to go and read who are the people who developed the technique and why this approach was considered to be a Nobel Prize award winning, discovery or invention, whichever way you look at it. So that pretty much brings the end to the, the third week of lectures, wherein we started looking into the various forms of the mutations, the so-called loss of function mutations, gain of function mutations, discussed issues with regard to why certain mutations are more frequent in the population, and how complex the gain of function mutations are, and how do you model organisms to understand human genetic disorders. So with that, we end third week. The last week lectures, that is week 4 lectures, would be devoted to discuss how you identify the disease gene. So you know only a disease that runs through a family. So how will you identify which gene is defective? So the gene discovery we are going to discuss, both for monogenic form and the current approach to identify the genes responsible for so-called polygenic disease or complex disease. How do you know what are the genes that are risk factors? So that would be the last set of topics that we'll be discussing in this course in the coming week.